Hello future doctors, welcome to Quick NEET Botany. In this video, we are going to continue the discussion of hidden lines of NCERT from the chapter Molecular Basis of Inheritance. Today's topic is genetic code. There is a line in this genetic code topic. The process of translation requires transfer of genetic information from a polymer of nucleotides to form a polymer of amino acids. Here, polymer of nucleotides, this is nothing but mRNA molecule. Polymer of nucleotides, ribonucleotides these are. And here if you see, the polymer of amino acids, that is a protein. So the simple meaning is translation means the transfer of genetic information from mRNA to a protein. And in this process, neither does any complementarity exist between nucleotides and amino acids, nor could be drawn theoretically. Let us take simple explanation for this. If it is mRNA molecule, on this mRNA molecule, suppose these are nucleotides. AUG is a nucleotide sequence, and if you see here AUG, here UUU, here UCA, suppose these are the sequences on mRNA. You know this AUG is coding for methionine amino acid. UUU is coding for phenylalanine. UCA is coding for serine. Of course, these nucleotides 3 together coding amino acids, that is genetic code. We are going to discuss about this now. But is there any complementarity between AUG and methionine? No. Is there any complementarity between UUU and phenylalanine? No. At the same time UCA and serine? No. And also we cannot draw it theoretically. It is a procedure. We cannot draw it theoretically. It is not possible to draw it particularly theoretically. And also no complementarity. Next. George Gamow, he proposed that genetic code is a triplet codon. You know there are 4 types of nucleotides in RNA. Uridylic acid, cytidylic acid, adenylic acid, guanylyc acid. So these 4 types of nucleotides, 4 cube. If these are triplets, they are forming codons with 3 nucleotides respectively, then it generates 64 codons. 4 into 4 into 4, then 64 codons. You know that out of 64, 61 are coding for amino acids, remaining 3 are not coding for any amino acids. 61 codons are sense codons and remaining 3 codons are nonsense codons. And to provide the proof that codon was the triplet, there are so many scientists' contributions. Let us see Hargobind Khorana. He was instrumental in synthesizing RNA molecule with a defined combination of bases, which are homopolymers and copolymers. What is meant by homopolymer? Let us see. I am taking polyuridylic acid. UUU, 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 like this. Homopolymer means in genetic code it is a triplet codon, right? In that triplet codon all the 3 nucleotides should be same. That is homopolymer. Here 3 codons we have seen. UUU, UUU, UUU. 9 letters are giving 3 codons. But we don't know in the beginning. They are giving all phenylalanine molecules only. one UUU coding for phenylalanine, another one is coding for another phenylalanine, and again one more phenylalanine. 9 by 3. That is equal to 3. 9 nucleotides by 3 phenylalanine molecules. 9 by 3 is equal to 3. So code is a triplet. Haragobind Khorana proved this. What is meant by copolymers? Two or more than two different types of nucleotides. Here I am taking UG. Several times I am taking UG. 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 Like that. See. UGU. This is one codon. GUG. One more codon. UGU. One more codon. If you take this. This UGU is coding for cysteine. This GUG is coding for valine. This UGU again coding for cysteine. Suppose here if you write GUG, it is coding for again valine. Here also if you see 1, 2, 3, 4 codons, 12 nucleotides are there. They are coding for 4 amino acids. 12 by 4 is equal to 3. This is also proving that genetic code is a triplet. Next. What are the contributions of Marshall-Nirenberg? Marshall-Nirenberg's cell-free system for protein synthesis finally helped the code to be deciphered. He did not use cell. Without using cell, he synthesized the protein in the test tube, cell-free system of protein synthesis. It helped to decipher the genetic code. He used this enzyme, Severo-Okhaya enzyme. The scientist Okhaya, he first observed this enzyme, hence that enzyme was so named, Severo-Okhaya enzyme. Its common name is polynucleotide phosphorylase. By using this enzyme, he deciphered the code without using the DNA template. It was also helpful in polymerizing the RNA. First, RNA synthesis is needed for protein synthesis, no? So, he polymerized the RNA with defined sequences in a template-independent manner. Without using the DNA template. Without using the DNA template. Template independent manner. Without using the DNA template, he synthesized RNA. By using enzyme, polynucleotide phosphorylase enzyme, Nirenberg, he used this technique. So this RNA synthesis is mainly based on the enzyme. It is not based on the DNA. So this is also called as enzymatic synthesis of RNA. These are the contributions to decipher the genetic code. Let's see what are the 64 codons. How can we remember? So you can write this UCAG this side, UCAG this side, UCAG this side, and for every box, for every row UCAG we can use this one here also. You know, you can write this. This is a checkerboard just like a dihybrid cross checkerboard. But how can we remember? Here I am writing the sequence for our convenience. I think it may helps to remember the code. Let us see. What is the first letter? U. The first letter is U. So again U here I am writing. U with U. In this UU sequence, we can write Phi square, Lu square. Phi, phenylalanine. The first two, UU is the sequence, no? First two letters. First letter is U, next letter is C. So UU, U, this one. And UUC, this one. UUU, UUC. These are coding for phenylalanine. Next two. Third letter is A, fourth letter is G. So next two means UUA, UUG. That is leucine, phenylalanine and then leucine. So first box done. Second box let us see UC. The sequence is UC. All codons with UC sequence are coding for serine only. C. UCU, UCC, UCA, UCG. So SER4. Second box done. Third box. Let us see the third box. UA series. UA. First two. Tyre square, tyrosine square. Tyre means tyrosine. What are those first two? UA, U. UA, C. Tyrosine. Next two are stop codon. So stop square. Third letter is U. So third letter is A. So UAA. Fourth letter is G. UAG. Stop codons. So third box done. Let us see the fourth box UG. First two. These first two codons are coding for cysteine, cis square. UGU, UGC. And the third codon is coding, not coding for any amino acid. It is a stop codon. That is UGA. And the fourth codon is coding for tryptophan. That is UGG. Fourth letter is G, no? UGG. So we completed the first row. Second row C. The letter C. The first letter is U and again the first letter is U. All are coding for leucine only. Leu4, CUU, CUC, CUA, CUG. The next letter is, next series is CC. Again, CCU. CU sequence, if you see, all four are coding for leucine. And CC sequence, all four are coding for proline. CCU, CCC, CCA, CCG. So second box, second row, second box done. Third box, CA is the series. CAU, CAC. First two are coding for histidine, his square. Next two coding for glutamine, GLN, glutamine. CAA, CAG. So CAU, CAC, histidine, CAA, CAG, glutamine. Next, CG. All four are coding for arginine. CGU, CGC, CGA, CGG. So second row done. Third row. AU. AU. First three. AUU, AUC, AUA, AUG. First three codons are coding for isoleucine. ILE stands for isoleucine. ILE3. The last codon AUG, that is coding for methionine. AUU, AUC, AUA for isoleucine, AUG for methionine. So first box done. The second box AC. All AC codons are coding for threonine, THR4, ACU, ACC, ACA, ACG. Second box done. Third box AA. First two coding for aspargine, ASN aspargine. Next two codons code for lysine. First two AAU, AAC. Next two AAA, AAG. Third box done. Fourth one AG. First two code for serine. Next two code for arginine. First two AGU, AGC. Next two AGA, AGG. So last box also done in the third row. Fourth row. First box GU. All GU codons coding for valine, Val4. GUU, GUC, GUA, GUG. Next, all GC codons are coding for alanine, alanine, alanine 4. GCU, GCC, GCA, GCG. GA, first two coding for aspartate. First two are coding for aspartate, ASP, aspartate, ASN, aspargine. Next two codons coding for glutamine, glue square. So GAU, GAC, aspartate, GAA, GAG, glutamate. Next, the last one, GG. All four are coding for glycine. So, gly4, GGU, GGC, GGA, GGG. So, this is a technique, at least it helps you to remember the genetic code. And out of these 64 codons, 3 codons are here. You know the properties of genetic code. Code is non-degenerate, code is degenerating, code is unambiguous, code is universal, of course. But here serine, leucine, arginine, they are exhibiting maximum code degeneracy. They are exhibiting maximum code degeneracy because these 3 amino acids are having more than one codon, 6 codons. They have 6 codons. Tryptophan and methionine, no code degeneracy because they are coded by one codon only. There is no code degeneracy for these 2 amino acids. Next. Classical example of point mutation is change in the single base pair in the gene for beta globin chain result in the change of amino acid residues glutamate to valine. If you see, GAG is glutamic acid. So the DNA sequence which is coding for this is CTC. This is on template strand and this is on coding strand. So if you see, that is the DNA sequence. The DNA sequence is coding for this amino acid. GAG codes for glutamate. It is nothing but human hemoglobin, one of the amino acid in the beta globin chain of human hemoglobin. Suppose in the place of this C, if it is replaced by A, T is replaced or mutated to A, CAC is giving G, A is complementary to A is U. G, here also it is changing GTG, right? Then it is forming valine. So in the place of glutamic acid, because of the change in case of single nucleotide, it is coding for valine in one of the beta globin chain. Then sickle shaped red blood cells, that is nothing but sickle cell anemia. Next. Here in case of insertion, deletion, frame shift mutations, insertion or deletion of one or two bases. This insertion or deletion of one or two bases change in the reading frame from that point of insertion or deletion. If you see, we inserted B. Before that, actually the statement is Ram has red cap. But here it has changed as Ram has BREDCAP. From this point of insertion, reading frame get altered if we insert 1 or 2. In the same way here also, reading frame getting altered. At a time, if we insert 3 letters, after the point of that insertion or deletion, reading frame is same. Before also it is red cap, now also it is red cap. That's it. Next. The secondary structure of tRNA has been depicted that looks like a clover leaf. The secondary structure of a tRNA is clover leaf. It's a 3D picture or it's a 3D structure. Actual structure means 3D. It's actual structure or 3D structure. We can say this is 3D structure, 3-dimensional structure that is looking like a inverted L. Very important points. Definitely you are going to get confusion in this. So do remember, secondary structure is looking like a clover leaf and the 3D picture or actual structure is looking like a inverted L. Next. Here there is a line in translation. The first phase itself, amino acids are activated in the presence of ATP and linked to their cognate tRNA, and this process is known as charging of tRNA or amino acylation of tRNA. In the first phase of translation, it is going on. But actually this is not the original beginning of translation. The first phase of translation is nothing but Amino acids are activated in the presence of ATP and linked to their tRNA molecules. This is charging of tRNA or amino acylation of tRNA. It is happening before translation as the first phase. In the second phase, original translation takes place. In that, when smaller subunit of ribosome encounters an mRNA, the process of translation of mRNA to protein begins. This is the original beginning of the translation, and do remember, this is the previous NEET question. When the smaller subunit encounters an mRNA, the process of translation, it begins. And here we can see in translation, to form the peptide bonds, RNA acts as the catalyst, that is 23 S type of ribosomal RNA in bacteria and 28 S type of ribosomal RNA in eukaryotes. In eukaryotes, it is 28 S type of ribosomal RNA. This is called peptidyl transferase or ribozyme. And if you see, a translational unit in mRNA is the sequence of RNA that is flanked by the start codon and the stop codon, encodes for a polypeptide. Here, the portion on mRNA, if it is that mRNA, which is the portion which is located between the stop codon, between the start codon and stop codon, particularly this portion is known as translation unit. Sometimes to confuse the student, they'll give, instead of start codon, they'll give promoter. Instead of stop codon, they'll give terminator. That is wrong. Suppose if the statement is like this. In a translational unit, the sequence of RNA that is flanked by the promoter and the terminator codes for a polypeptide. Wrong. That is flanked by the start codon and the stop codon. It is coding for a polypeptide chain, or it is coding for a polypeptide or protein. And what is the location of UTR regions? You know already. UTRs, which are the regions on mRNA which are not translating but they stabilize the structure of mRNA and they are required for efficient translation process. These are present before the start codon and after the stop codon. Don't get confused. After the stop codon, before the start codon. Wrong. Here before the start codon, after the stop codon. Before this, after this. These are UTR regions. In this process of translation, amino acids are added one by one, translated into a polypeptide sequences which are dictated by DNA and represented by mRNA. Here DNA is the director of the movie and mRNA is the hero. So in the process of translation, the amino acid sequences in a polypeptide chain are dictated by DNA. It is giving genetic information, that is playback material. And who are playing that role? mRNA. Simply you can remember this point. Next, regulation of gene expression. In this regulation of gene expression, it occurs, that control of regulation of gene expression, it occurs at 4 levels. Here it is given. Transcriptional level, because protein synthesis, protein is responsible for the expression of character. It should be under the control, the synthesis of that protein. That control, it occurs at any of these 4 levels in eukaryotes. These 4 levels are transcriptional level, processing level. You know that in transcription, hnRNA formation, that is primary transcript. Later hnRNA is converting into mRNA, that is processing level, splicing, we can see in that. And the third one is transport of mRNA from nucleus to the cytoplasm. And then finally translational level. So it occurs at any of these 4 levels, majorly at this level. This is the major site, major target initiation site of that control of gene expression. It occurs these 4 levels in eukaryotes, of course, 4 levels in eukaryotes. But in prokaryotes only this one and this one. First and fourth one in prokaryotes. Why? Because in prokaryotes, no need of any processing, no need of any transfer of mRNA to any other site. Both transcription, translation, they occur at a time and simultaneously in the same compartment, that is cytoplasm. So in eukaryotes, it occurs at 4 levels. In prokaryotes, it occurs only two levels. Those are first and last. Next. Here, if you see this regulation of gene expression, it is under the control of metabolic, physiological or environmental condition. They are regulating the expression of gene. Lactose is there in the medium in case of lac operon. We are going to discuss next. So that is environmental factor. Metabolism, lactose, it is a destructive mechanism, and that metabolism is a physiological mechanism. So all are regulating the expression of gene. Generally, in most of the organisms, in prokaryotes also, the control of rate of transcriptional initiation is the predominant site for control of gene expression. Here we are taking lac operon. In case of lac operon also, we are discussing about the control of the rate of transcriptional initiation, only the predominant site. But when you go to RNA interference of biotechnology, that is at translational level. Here lac operon at a transcriptional level. And here there are certain regulatory sequences. No, these are accessory proteins. They may express as a positive proteins, activators, or negative proteins, repressors. If they are positive proteins, that is positive regulation as well. That is beyond our discussion. If these are negative proteins, those are repressors. We are going to discuss about the repressors. Here there is a statement. The accessibility of the promoter regions of prokaryotic DNA, as in many cases, regulated by the interaction of proteins with sequences termed as operators. If you see this promoter and operator, this promoter, it has to accept RNA polymerase. This promoter, it has to accept RNA polymerase. The accessibility of that promoter to accept the RNA polymerase, it is based on the interaction with the operator. So operator is deciding the promoter capacity to accept the RNA polymerase enzyme. And in lac operon, if you see, there is a statement. Y gene, which codes for, actually Z gene for beta-galactosidase enzyme. Z gene codes for beta-galactosidase. Y gene for permease. This permease increases the permeability of the cell to beta-galactosides. Here beta-galactoside means lactose. That is not beta-galactosidase. That is not beta-galactosidase. Here beta-galactoside, if you see that word clearly, you can understand. Beta-galactoside means lactose. Permease increases the permeability of the bacterial cell to take up that lactose. Next. Here one more statement we can see. Remember, a very low level of expression of lac operon has to be present in the cell all the time. Because permease is a bit required in the beginning for the entry of that lactose into the cells. So to produce that permease continuously, that very low level of lac operon expression should be needed in the cells to produce the permease. Then only lactose can enter the cells. Otherwise lactose cannot enter the cells. And if you see, the repressor of the operon is synthesizing. See, if you see, here also repressor is producing. In this situation also repressor is producing. Continuously that repressor protein is producing by that I gene. So this is constitutive gene. I gene is constitutive. Continuously it is producing its product. Constitutive, or we can say housekeeping. Housekeeping wala always they can do their duty, no, in the same way. If you see ZYA genes, if you take these ZYA genes, here they are not producing. In this case they are not producing, but here they are producing. So whenever they need, then only they are producing. So these ZYA genes are non-constitutive genes. Non-constitutive, or we can say luxury genes. These are non-constitutive genes or luxury genes. Next. Regulation of lac operon can be also visualized as regulation of enzyme synthesis by its substrate. Here lactose is the substrate, right? Lactose or yellow lactose, majorly yellow lactose or secondarily lactose. Generally, do remember this point. Majorly yellow lactose is the substrate, secondarily lactose. So if that lactose is the substrate, it's a synthesis, it's a destruction. What is the destructive enzyme of lactose? That is beta-galactosidase. Beta-galactosidase synthesis induced by whom? Lactose only. So regulation of lac operon can also be visualized as regulation of enzyme beta-galactosidase synthesis by its substrate, lactose only. Lactose only responsible for its destructive enzyme. And the last point here we can see, can you think for how long the lac operon would be expressed in the presence of lactose? Till that lactose completely consumed. Do remember this point. This is the answer. Till the lactose in the cell completely consumed. Till the lactose completely utilized or consumed. Till that period, lac operon can be continued.